Yeah, okay. My disclaimer is I'm not an expert in risk assessment, risk management policy. As a molecular toxicologist, I'm just really interested in understanding mechanisms at the cellular level of how contaminants may or may not cause adverse effects. And so what I thought I would do today was talk a little bit about nanomaterials in the environment and challenges we face as toxicologists of assessing their unintended exposures. So I'll talk about nanoparticles that will be released into the environment, and how those types of exposures may have a toxic impact on risk. I'll try to talk a little bit about the influence of public perception on science, and then I'll talk somewhat about some of the data that we've been collecting at University of South Carolina as a team of researchers studying environmental toxicity of these particles and some of the unique and unexpected effects that we're finding. Okay. So Vicki showed you a graph yesterday of the exponential growth that we're going to experience in the use of nanomaterials over the next decade. So because of this growth, it's inevitable that these compounds are going to end up in the environment. And so one of the tasks we have as environmental toxicologists is understanding what the risk of exposure to these things will be. So this is exposure in a way that these compounds were not intended for. So, and they'll be released from a variety of sources, and as someone alluded to earlier, I think it was Jennifer, that we don't even know what these sources are at this point. Okay, but there are going to be a variety of sources. There's going to be a variety of uses and commercial uses, consumer product uses, but what happens when the environment interacts with these products? So you have a commercial product and it's exposed to heat and light and so forth. What happens to the nanomaterials in those products? Environmental fate and transport. There we go. When these particles get out into the environment, where do they go? Do they change from a chemical standpoint? How do these changes impact biological effects? Do they get dispersed over long ranges or distances? Or do they stick in sediments and stay close to the source of input into the environment? And then we have this whole aspect of what happens during recycling. So what happens to a nanoparticle through its life cycle, if you will? And does the form of the particle change, and does that get reintroduced back into consumer products somehow, or say drinking water or so forth? And how does the exposure and risk of those change? Okay, so it's not a question of if, but we're now facing the questions of when they get to the environment, what are they going to do? Okay. So there are a variety of challenges in accessing environmental exposures, but the one that I'm really going to focus on is complexity, and so we just heard about that with respect to the risk assessment and policy process. But before we even get there, we have this super challenge of this complex network of the ecosystem. Okay, so just the mere tracking of a particle through food webs and dealing with bioaccumulation and biomagnification and which species are sensitive and which species are not sensitive, and trying to understand that is very complex. Where these complex materials go, do they interact with one another in the environment, and does exposure to mixes of these contaminants pose different kind of threats than they would on an individual basis? What are the effects from a water exposure versus an air exposure versus a skin exposure? Okay, so all these things constitute an extremely complex network that we as a molecular toxicologist try to piece apart. And then certainly what we're here talking about is the impact that public perception has on the science we're doing, and the impact that we do as scientists have on the public perception. Okay. So as an example, so you have nanoparticle X and it moves from a consumer product into soil and into groundwater. If I asked each one of you a scenario of what do you think could happen to that particle and how it may impact drinking water, you probably all come up with a different scenario, okay? So you have a carbon nanotube and it forms big aggregates, and so it blocks up pores in wastewater treatment facilities, okay? You have nanoparticles that get out into the environment and interact with other contaminants, and so it changes the way those are removed from wastewater treatment plants. Okay, and these scenarios go on and on and on. So not only do we have the complexity starting out where Vicki Colvin was talking about, where we have so many particles and they have all different charges and shapes and they're chemically modified and they have different toxicities. Now we're talking about what happens with each and every one of these things when they get out into the environment, and you have all these other confounding factors on top of that. Okay, so how do we begin to study the safety of nanomaterials as bench scientists? And this was a paper that came out in 2006 by Andrew Maynard and colleagues, some of which are here. And they came up with 5 grand challenges we face and how we should address them. And they go through these challenges and talk about implementing new technologies and sensors. So how do we detect these things in the environment, and that type of aspect is needed. Coming up with models for predicting engineered nanomaterial behavior in the environment, and modulating policy and so on and so forth. But one of the early challenges that they talk about that we face is communication, and communicating research on nanotechnology risks and benefits, not only to other scientists, but to the public as well. And so obviously this communication process is important, both for the public and how they perceive things, and also for us as scientists. So we've been talking a little bit about what do we do as bench scientists, and how do we take the data that we gather and relate it to risk. And Vicki talked about a more modeling approach, and so trying to put input in and have come up with some kind of prediction on how can we predict risk, okay? But that also needs to be still complex with just basic fundamental knowledge of environmental impacts of nanomaterials. And we need to understand both how the effects of the environment, what the effects of the environment have on living systems of nanomaterials, and what nanomaterials have and their impact on the environment. Okay, and then of course all of this will feed into risk assessment, which Jennifer just talked about. So last night when I was thinking about some of the things that have been said, I was thinking, what is my responsibility as a bench scientist in this whole risk communication process? And does what the public think really influence my research and what I do? And so I think there's a variety of ways that the public does. And one of those is by money. So I think funding availability is directed by what the public thinks about contaminants, and also that falls into and influences risk, as we just heard about. But so if you take bisphenol A, for example, so this is something that's resurfaced over and over again. So bisphenol A is as a plasticizer, and it's getting a lot of press lately because of its use in baby bottles. Okay, and so endocrine disruption was really hot, about 10 years ago, and then it sort of died down. But now there's sort of this resurface, and I've seen some funding available now to look at things like bisphenol A. And interestingly enough, it used to be very difficult to get bisphenol A-free baby bottles, and now you walk through the aisles of Target, and there they are. Okay, so I think public's perception can have a great impact on us as bench scientists. So something else I was also thinking about, and these are sort of just my thoughts of what I've gathered through the last day of talks. So I may be way out of line here, but if you look at some of the things I'm working on, I'm looking at lung epithelial cells, and I'm looking at exposures to carbon nanotubes. Okay, and as a mechanistic toxicologist, I'm really interested in which genes are turned on and off. And so we've done microarray studies, and so we expose these cells to carbon nanotubes, and we see, okay, all these genes are upregulated and all these genes are downregulated. And I was thinking about presenting this, and I said, well, I could stand up here and say that all these gene changes produce a unique signature to carbon nanotubes. But I could also say that a number of the genes altered by carbon nanotubes are similar to what we see with asbestos. And so we've been talking about this framing